All right, well, welcome to this most recent episode of the Akkad and Coco Report. Today we have the pleasure of having Bobby Yeh on. Dr. Yeh is a cardiologist and a clinical researcher at the Beth Deaconess Medical Center. He is he's one of the few people that I think I've found that has incredible amounts of depth from a clinical standpoint, but is also, does spend such a tremendous amount of time clearly doing research. He's here to talk to us about what I kind of think is like meta research. He's looking to try to see on his most recent publications whether or not there's a better way to come to the answers that we all seek. Everyone wants to know whether or not a device is safe. Everyone wants to know whether a certain drug may be effective. And traditionally, this has been done via randomized controlled trials, at least for the recent, that's the current dogma, that you have to have randomized controlled trials to really figure out efficacy and safety. That, of course, can be a very expensive proposition. It's about $1 billion. I think there's an estimate that says there's about $1 billion to bring a drug to market, and that automatically should make a lot of people very concerned, because if that's the barrier to entry of bringing drugs to market, you got to be really concerned about what drugs aren't coming, or what devices or what other therapeutics are not coming to market, given how expensive it is to do randomized trials. So, Bobby, what are your thoughts on a path forward? Certainly based on your research thing.
Well, thanks for the introduction. Thanks for having me on, Anish. It's good to be back, and it's great to get a chance to talk about some of the work we've been doing. We have a lot of thoughts on this issue, as do I think many people, and there are many groups that are working in this area of trying to basically better understand whether or not the therapies we give work. And as you mentioned, the gold standard of randomized controlled trials, of course, are not infallible, but certainly the highest level of evidence I think that we can provide to get causal estimates of whether or not things work. But they're extraordinarily expensive. They are often hard to conduct. And as a result, I think you see so much of the published literature and so much movement toward this idea of, well, what can we understand from real world data? And also in part because randomized controlled trials traditionally don't enroll a diverse and representative population. So there are many reasons, although they are very good scientific experiments, that there are many reasons why randomized controlled trials may not give you the precise answers to the questions that you're looking for. And many have turned to real world data and real world evidence to find those answers. And actually, as you know, it's as part of the 21st Century Cures Act passed in 2016, there's actually a explicitly stated movement toward trying to use real world evidence to better understand even regulatory decisions. So the US FDA has a comment on their website about adopting really a new approach to understanding whether or not things work and are safe using real world evidence. So, but I think if there's anything that we've taken away from research during this time period of COVID, is that observational research methods with real world data are challenging and fraught with potential problems. And of course, we've known this long before COVID. I think COVID has somewhat accentuated and certainly brought it into the public sphere more about how hard observational research methods and study designs can be. And so what this group of studies that we've tried to do most recently that you've brought me on to talk about is try to bridge this gap between real world evidence and randomized controlled trials. And namely what we did was really try to see whether or not real world evidence could replace the outcomes. So not so much replace the randomization. I think randomization, many people consider, is magic. It really is one of the greatest modern inventions, scientific inventions, is this idea of flipping a coin and assigning a patient randomly to a treatment assignment. So exploiting randomization, but then instead of following those patients with very expensive traditional CEC adjudicated, site reported outcomes, it's a very labor intensive effort for people who do clinical trials. Instead, seeing whether or not real world data in terms of, in this case, administrative claims data from Medicare could actually sort of match those outcomes that were ascertained by CEC adjudication. So that's a long-winded answer to sort of set up the problem that we tried to tackle with these latest studies.
That's great. And it's particularly relevant to, should be particularly relevant to cardiologists because, as I mentioned, the cost of bringing it's commonplace for the cost of bringing a cardiovascular drug from discovery to regulatory approval. That cost exceeds $1 billion. And as you're saying, most of that cost resides in the conduct of clinical, in how we conduct clinical trials and how one clinically adjudicates the events. Correct. So you actually have. I actually didn't realize this till not that long ago. That you actually have sponsors of the trial send out folks to actually review each single chart to arrive at events and side effects. Is that correct?
Yeah, it's called site monitoring. When the finding of an endpoint in a cardiovascular clinical trial, this is what it takes to do. So you roll a patient in a trial. Let's say it's a stent trial or a drug trial, and then if it's something like a procedure, you captured that patient at their index procedure quite well. So that is sort of the easiest part of the trial. It's not easy to enroll patients, but once you have that patient enrolled, it's easy to collect that information that determines, what stent they got and what were their characteristics. You have them there in front of you. You consent them, you enroll them, but then they go home, and then you have to follow those patients for an assortment of outcomes. And while you may have scheduled visits to reach these patients, sometimes these are in person or sometimes they're telephone visits. Sometimes the patients don't show up. They won't answer their phone. They move. They didn't give you that information. And then when you ask them the questions like, well, in the past 6 months or whenever it is since I last saw you, did you get hospitalized for any problems? And sometimes they can't recall, or they tell you something that may not be, maybe inaccurately recalled. So that becomes very difficult. And so what a site has to do then is, let's say there was a hospitalization for a heart attack at another, not your index facility, but a different facility, then the site coordinator has to go find those records, and they've got to get those records, and then they fill out something, and then they send those forms in to a centralized adjudication committee, because ultimately it's this centralized, maybe blinded adjudication committee that has to determine whether or not whatever happened to that patient met the criteria for a true heart attack. That's a committee of 5, 6 physicians applying a stringent criteria in a blinded fashion. That has to be coordinated by usually, academic or clinical research organizations. You can imagine, I've just went through so many different steps to get one endpoint for one patient, and you just magnify that, scale that up to a 5,000, 10,000, 20,000 patient clinical trial, and you can see that the costs quickly add up in terms of the manpower, and the fidelity may not be always perfect either. So it's an extraordinarily important and expensive endeavor that happens. It's getting more expensive. And exactly what you said about this is important to cardiovascular. We've sort of shot ourselves in the foot. We've done too good a job. Cardiovascular events are on the decline. Event rates used to be, say, 10%. They're now 5%, 3%, 2%. We treat these patients so well, and with lower event rates means that any new drug or device that's going to show benefit has to enroll that many more patients. And so we're talking about this multiplier for the cost of the trial. So I think there are a lot of people, patients, physicians, industry, everybody has a vested interest in trying to understand how we can make clinical trials more efficient so we could better evaluate therapies, do larger powered studies, do them more quickly, get studies for devices and drugs that are understudied. We need to make this process more efficient. So that's why we're doing some of this work.
And so we have this, we do have this other repository of information, and that's the claims database, which kind of exists mostly from a billing standpoint, right? So you have these diagnoses codes that are put out that every single physician knows about, and those, and in order to bill patients, in order to bill payers or bill Medicare or whatnot, you have to enter those codes in. And so we have these giant claims databases that exist. So, the overall, so what you folks did, you took 2 different studies. The one study looked at the U.S. core valve high risk study, the SURTAVI, SURTAVI study, correct? And tell us about how. So that was basically a randomized control trial that was done looking at transcatheter aortic valves. And you have a nice little data set there that shows efficacy, safety, all these endpoints. And and we also have this claims database that also exists. And so somewhat ingeniously, in terms of looking to see whether or not you can get the same data that you got from the randomized control trial that was super expensive to do. Your mission, as I understand it, was to look at look and find and see if those same patients that were in SURTAVI could be linked to the Medicare claims database, and would you be able to kind of follow them and get the same results as the RCT?
That's right. The trials that we looked at two. They were the high risk pivotal and the SURTAVI transcatheter aortic valve replacement trial. So for those not familiar, this is a TAVR device. It's a non-invasive way to replace an aortic valve, and they're really, at this time, certainly early on, there was kind of two primary valves that were used, and this is one of the two primary valves that were used. The core valve device is a device made by Medtronic, and really these were, I think, groundbreaking, very expensive, but very revolutionary randomized clinical trials that really have, I think, shifted the entire paradigm of how we treat aortic valve disease. These were regulatory IDE, investigational device exemption trials for the first approval of these devices in two different populations. So the high risk patients and then the intermediate risk patients. And exactly right. What we tried to do is take those exact patients that were enrolled in this randomized trial and find that patient who was enrolled in the trial, find their medical records in Medicare claims. And it just so turns out that Medicare treats patients, covers patients who are 65 and older for the most part. And transcatheter or aortic stenosis, the disease that we're talking about, tends to afflict elderly patients, many of whom are insured by Medicare fee-for-service. And so this is a good way to do this. We don't have a national registry system that covers all payers in this country like they have in other countries. Medicare is probably our closest thing that we have that covers really a broad swath of patients in this country. And so we found these precise patients in those trials within Medicare claims, and then just tried to compare, had we followed these patients with claims in lieu of the clinical trial sort of event adjudication machinery, would we have met the same kind of p-values? The hypotheses that were tested would they have been answered in the same way?
And what did you find when you?
Yeah, so the primary hypotheses of those trials, the primary questions being asked were, the primary endpoint was mortality for the high-risk pivotal study. For the SURTAVI study, the primary endpoint was the combined stroke or death. And we found for those two endpoints, the clinical trials and the claims found almost identical. For the mortality alone, it's identical. I think registries and administrative claims find deaths with equivalent, if not greater, precision than clinical trials can. Because when a patient dies, that information goes into the Social Security and their Medicare benefits, et cetera, are known. So those are very precisely identified. And then for an event like stroke, we sort of published this separately earlier, but stroke, not perfect, but pretty good. And in this case, where strokes may differ from adjudicated strokes and claims-based strokes, again, it's a randomized study. So you think that the errors that might happen would be evenly distributed between the two randomized arms, so it wouldn't create a bias. So in fact, that's part of the reason why we think that claims can be useful in the randomized setting, because it's not anticipated that where claims might be inaccurate, they would be inaccurate preferentially in one treatment arm versus another. That may not always hold, but at least for these endpoints, that appeared to be the case.
Yeah. Were you able, so this required linking the two databases, you had to find the same folks in SURTAVI, RCT,
Yeah.
you had to find them in the Medicare claims database as well.
Yeah.
And I know, of course, this sets up nicely because a lot of folks who get TAVRs are over 65, as you said. How, just in terms of how successful this could be in the future with Medicare claims database, what percentage of folks were you able to link up?
Yeah, we linked, it's about 2/3 for one of the studies and about 80% for the second one. One of the challenges here is that an increasing number of patients are insured through Medicare Advantage. So we don't have Medicare Advantage, at least we didn't at that time have Medicare Advantage claims, but Medicare Advantage claims have since become available, actually. And we're starting to do some work with MA, Medicare Advantage claims. So you can see if those claims work out well, then there's going to be the opportunity to link virtually all patients over 65 years of age in the US. But for other disease processes, you're not so lucky where it's all 65 and older patients. As TAVR moves into lower and lower risk patient populations, there's a chance that you're going to get some 50 or 60-year-old patients and they're not covered by Medicare. And actually, in the other sister publication for this one, both of these were published in the journal Circulation. The DAPT study was another study that we tried to do the same thing for. So the DAPT study is the dual antiplatelet therapy study. It's a comparison of different durations of dual antiplatelet or blood thinning medications after patients underwent coronary stent procedures, heart stents. And in this case, a large number of patients could not be linked. So we linked, I think fewer than half of the study. And so for that population, which was patients who were undergoing heart procedures for coronary artery disease, the distribution was not quite as favorable. So, this is an imperfect approach. One can imagine that, you could preferentially do studies and you exploit this approach in older patient populations. Atrial fibrillation would be one that you could probably do this for, and the disease processes of the elderly. But right now in the US, there isn't a great kind of all payer across the age spectrum system where electronic health records or administrative claims are captured.
Since you had fewer patients, or I guess you end up with fewer patients for a variety of different reasons. Does that does that change the does that change how likely it's going to be that you'll come up with a result that matches the original trial, I guess?
Yeah, I mean, I think it depends. When you lose patients for a matching like this, then your first question is, well, are the ones we couldn't match different than the ones we could match?
Right.
There's one obvious reason why they can't match, and that's age. So they're not insured by Medicare. So that's something that's knowable. But there might still be patients that you can't match for whatever reason because you can't find them, even though they look like they should be matchable. That's another reason. And then you really want to determine whether or not you've created some sort of systematic bias by comparing those two patient populations. And luckily in the trial, you have all the baseline characteristics so you can compare them. But, people look at this and they say, well, if you're going to miss that many patients, and that is effectively reducing your sample size. And so you've lost out on some of the things. But, this is just one thing that we're doing retrospectively. If you were to do this prospectively, you wouldn't do this deterministic match like we're doing. You would just collect the social security number for the patient at the time of enrollment as a data point, and then you would be able to perfectly match that patient. So let's say if you wanted to do this prospectively in a TAVR population, you would say, well, I'm only going to enroll Medicare insured patients, fee for service, say. You would know that if they were privately insured or they were Medicare Advantage at the time of their procedure, and if that would just become an enrollment criteria for your trial. And then you would capture that information, which would allow you, a date of birth, a social security number, whatever it was, that you can send directly to CMS. Actually, it's an organization that works with a partner with CMS. They are used to receiving that type of information for linkage. So if you collect that information prospectively, you would have a much better match rate. You could easily have a perfect match rate or a near perfect match rate just by specifying the population, collecting the right information at baseline.
I see. So this, because of how exactly you linked from one database to the other, I didn't quite understand, because obviously you don't have unique identifiers that you collected probably from a core.
Yeah. We didn't use unique identifiers. That can be done. So, the transcatheter valve therapy registry, the TVT, the STS ACC TVT registry. That is a registry that is linked to CMS claims, and it's linked through direct identifiers, social security number, dates of birth. So that you have that sort of information. So that can be done. That's not what we use because we don't love handling that sort of data here for all sorts of privacy reasons, et cetera. But what you can do instead is, there's a workaround, which is we know how old the patient is. We know the facility where they had a procedure, and we know that they had a TAVR, and those have a specific ICD-9 or ICD-10 code. And it turns out that there aren't many patients who are the same age, the same gender, at the same hospital who get a TAVR on the same day. And so the linkage works pretty well for a procedure like TAVR. That's one of the reasons why the procedure, it's a little harder to link for DAPT for PCI, because it's a more frequent procedure than TAVR. So you could have, 5 to 10 PCIs at a facility and only 3 TAVRs or 4 TAVRs.
Right.
So it makes it a little bit harder to link.
So it seems like this works pretty well. It seems that it works pretty well as long as you have a database that's comprehensive enough that encompasses things, correct?
Yeah. Yeah, and obviously this is best case scenario. We've done this in this particular trial because we happen to have this particular information available to us. So you can imagine a whole host of questions that you might not be able to answer using this approach.
This is a proof of concept in terms of going forward. But isn't the fundamental thing here is that you had an RCT, meaning you had you had a randomization surrogate or an instrument or what have you, right? So how does one, I guess, would you, I mean, and we've seen a lot of the problems with observational analyses that fundamentally are trying to do the same basic thing, or everyone's trying to do the same basic thing. Everyone's trying to come up with some causal inference, some causal mechanism, to try to figure out whether whatever intervention you're doing is causing this. Now, if we accept that randomization is the best way to do that, how do you go about making sure that, if you don't have an RCT, how do you go about taking these large data sets and making sure you have the right surrogate and all these measured, unmeasured confounders are balanced?
Yeah, that's a much more difficult question. I mean, we did something that was easier here, which was we relied on randomization. So I think what you're asking is, so short of randomization, what kind of methods can we use to really understand causal inference? And obviously this is not only just one field, this is like multiple fields and multiple disciplines trying to ask these same questions. But I think among the groups that think about this, for better or worse, it is the medical community that does the most of it, and it's the medical community that does it the least well. It's actually kind of a bad combination, but social scientists do all sorts of this kind of work. Economists, as you know, do this type of work. Epidemiologists do this. But it's clinical docs, I think, really do the worst job at this.
Yeah, but Bobby, how do you know? How do you know that the social scientists are doing this so well? They have the most well-developed methods to do it, but I had. So what we're talking about is part of what we're talking about is things like econometrics, correct? So economists and social scientists have derived over the many decades multiple different ways and tools of trying to get answers from data sets when you don't have randomization, because in a lot of social policy you just can't randomize. You can't randomize people to poor housing versus good housing. Just not ethical. So part of the issue. So and I don't know if you probably we just last week we happened to have Edward Lemer on. Edward Lemer is have you heard of him by chance?
Tell me more.
His name is Ed Lemer. He's an economist. And he, interestingly, in 1983, we had him on a podcast about four days ago, or a week ago or so. I should have sent it to you to get time to listen to. But he's an economist who in 1983 wrote an article saying taking the con out of econometrics. And what happened in 1983? He was writing that in 1983 because social science trying to be harder and more empiric has been happening for like a century, right? And so when he was writing the article in 1983, there's a number of folks that were trying to quantify a lot of different things within economics from data sets. Regression, simple regression, other techniques, and to try to come up with answers to basic questions like does capital punishment result in is it an effective deterrent? Does it result in reduced crime? Now that's a really complicated question to answer, but there were economists at the time in 1983 were saying, yeah, we can answer this because there are some states that have this capital punishment policy, other states that don't have this capital punishment policy, correct? And they would do some to us today, some relatively simple correlation and say, aha, see, this is what's causing that. And Ed Leamer in his, what is I think in the economics world, a pretty fundamental paper, wrote, kind of poked, as you can imagine, many holes in this kind of simple correlation, right? And that was in 1983. And then in 2010, there were, it took that long, what we joked about in the podcast, for someone to actually write a paper trying to rebut it, saying that, all right, we finally have taken the con out of econometrics, and this is how. And what they talked about, and as you'll be, which is, this is of course no surprise to you, they talked about how, yes, econometrics was a little bit of, emperor with no clothes, but now we had figured out how to do it. And the way we figured out how to do it is with techniques that are common today. Regression discontinuity, instrumental variables. And so, this battle has been going on for some time, and where the soft sciences have tried to be more experimental, but they've been trying to get towards, Elon Musk, figuring out what it takes for, a rocket to land, right? And how did you eventually figure that out? There's all this theory, but then you have to do it, and when you do it, all kinds of things go wrong, and you keep figuring out and finally get to a rocket landing on some drone ship in the ocean. And there's still that tension to Dr. Leamer is a really nice, fascinating conversation. Still exists, and so he still points out that, look, people want to say that it's an instrumental variable, but I was like, I don't know, it sounds very very hard and this is a fundamental thing that you found, but in the end of the day, it's just a surrogate. And then the problem with pseudo-randomization that uses instrumental variables is that you have to make sure the instrumental variable is good, right? And you've pointed that out when you've actually critiqued, I know, economic and analytics that kind of touch our world and said, hey, this instrumental variable doesn't work, and therefore the answers you're getting aren't correct. Yeah. So I guess, my kind of issue with seeing econometrics kind of bleeding into medicine now is that they've been doing it for a while. They're obviously well developed, but whenever I see a bunch of them talking about the stuff, it's like, well, what is your how do you know that? How do you ever know in these things that you got it right or you got it wrong, right? Yeah. In the world you work in, certainly, there's, in medicine in general, you kind of get that. We've always had that as a backstop, meaning, unfortunately, patients are, end up being guinea pigs, and you end up in dead ends. And I find the social sciences kind of struggle with that. So how does one, in this particular case, you of course use the RCTs as somewhat of your gold standard. How will we know? How will we ever kind of figure out, if you're using econometric methods in medicine, whether or not we're getting the right answer or not? Sorry, it's such a long-winded question.
No, it's a great question, because as you alluded to, in many cases, we know when we've been right because we've done the real experiment, and so we know the truth. But in most scenarios where if this is really going to be useful, then we can't know the truth. This has to be what creates the truth or helps us better understand the truth, not did it match the truth and when did it miss. There was actually a paper just this week in JAMA Open for oncology trials. They list a whole bunch of sort of observational comparative oncology trials, and you saw this, and the majority did not replicate the true finding in an RCT. I mean, it's hard to know exactly the details of how they, what that means, because sometimes you study patient populations or the interventions are not exactly the same. But there are many good examples in cardiology where observational studies have led us astray. The one that I talk about the most is the one that really set me off in this field, actually, was when we were very concerned about the safety of drug-eluting stents in the 2006, 2007 time period, 2008. And this was right when I was doing my cardiology fellowship, and I was also doing training in epidemiology. There were papers published in the New England Journal which suggested that not only were drug-eluting stents safe, but they were life-saving, apparently life-saving, in observational studies. You know, they weren't life-saving in 2007, 2008. It was just that we were choosing to put their alternative, the bare metal stents, in patients who had a whole host of comorbidities or couldn't take dual antiplatelet therapy, whatever. They were confounded analyses. They were published in the highest impact journals. And they were wrong. And they were, and it gets back to something that I know you think about a lot. Well, how did you know they were wrong? Well, we sort of knew they were wrong because it lacked all plausibility. It was just not biologically plausible, clinically plausible that these could have been saving lives within a 3-month period, because it's just not how the mechanistic, it sort of, it violated the mechanistic underpinnings of the device that we were using. And so we were kind of using biology and plausibility to determine that a study was wrong. And so there becomes this tautology, and I know that you're well aware of, but then they were later subsequently proved to be wrong, because we've had a large number of randomized clinical trials since then that showed that there's no benefit or harm for mortality when we choose one stent versus the other. And so observational research was getting it wrong. Obviously, the Women's Health Initiative is one prime example where observational research findings got it wrong, and the properly designed randomized clinical trial got it right. Since then, sort of experienced epidemiologists have reanalyzed observational data for the women's health, for estrogen hormone replacement therapy, and can get an answer that gets closer to the Women's Health Initiative. But again, here's this thing where we know the truth and we're trying to move toward the truth and approximate it, as opposed to just actually finding truth in a prospective way with observational research. It does remind me, one of my good friends is David Cohen. I think you know him. He said, some observational studies are right. I just don't know which ones, is what he always says. I think that we can do better than that. I think that there are some methods that are just stronger than others for particular questions. And what I always say is this: I say that the determination of which methods are going to best answer a particular question is not a statistical question. It's a clinical one, because the person who makes the clinical decision to choose treatment A versus treatment B, those are the types of the physicians who know what are the necessary variables to include in the analysis, because they can tell you whether or not they're confounders. And we could have told you as clinicians and cardiologists in 2010 that if you compared a bare metal stent patient to a drug-eluting stent patient, those were entirely two different patients that we were talking about. Those are not comparable patients. But a statistician can't tell you that. They don't know. They don't make that decision. An economist is not going to be able to tell you that. It's the clinician who needs to tell you that. But most docs also don't know that it's the propensity score methodology that's very vulnerable to that, or regression is, but an instrumental variable is not. Most clinicians can't tell you what an instrumental variable is, or a difference in difference design, or regression discontinuity. This is not language that most clinicians speak. So there is this divide, and unfortunately, I think the best design studies require, fortunately or unfortunately, they require the input of both methodologists and clinical subject experts, and the coming together of those two groups is quite unusual, and it's rare, but it's necessary for the studies to be accurate. Now, there are other specific approaches that we and others have used. There's when you did your scientific, when you did your biology class in high school, we all learned to run these gel electrophoreses. I don't know if they still do that sort of thing, right? You stick things you grind things up and you stick them in a well and you apply some sort of current across it, and then things move in little bands, right? And when you did those gel electrophoreses, you always had two wells on the side. One was a positive control and one was a negative control. And the negative control, you put nothing in there. And if you knew that if a band showed up on that gel electrophoresis, that something went wrong with your experiment. That was your negative control. That equivalent exists in clinical research. And what we call those are falsification endpoints, or placebo tests. If you're concerned about treatment A versus treatment B, you can just throw in these endpoints that you know that treatment shouldn't affect. And if you see a difference in that endpoint, then something went wrong with your experiment, if that makes sense. So
Right.
that's one example. There's different sensitivity analyses that can be done. There's so many ways that you can make a more rigorous observational study, but we don't have like kind of a standard recipe for that. For clinical trials, people know it's blinding, it's concealment, it's randomization, it's like there's this cookbook for randomized clinical trials that have become, I think, the science of randomized clinical trials. For observational research studies, we don't have that same, at least there are these recommendations, but we don't have that same cookbook that the whole field knows about. And
Right.
any sort of person who's a little bit like okay with data and could get their hands on a data set and a statistical software program can run some sort of observational research analysis. It's not the same, where you can't do that with an RCT. So the quality varies so widely with observational research. As a result of that, you have no idea what to believe. There's bad stuff out there and there's good stuff out there.
Right. So the idea would be that if with the right tools, we can come up with answers that we can perhaps better trust.
Yeah, exactly. And it's the right tools combined with also the clinical input. So
Right. And it's so interesting you say that because I asked Dr. Lemer, so how do you tell me as an economist, what's your advice to a doctor in terms of how do we what prevents us from becoming quacks? And he said he said they have to be able to tell that you have to be able to understand that there's some compelling story when they tell you that this is causing this. So if you as a clinician don't believe it, if you as a clinician can't find a compelling story, the drug-eluting stent lowering mortality, that is not a compelling story. That's not a compelling story as to
yeah.
and you can understand why that is. So that's interesting to say that. So his solution is you guys both arrived at the same place. Both of you are saying you need clinicians with knowledge, with priors that are informed to do this type of research, so that you don't end up on some weird place.
But I was, it's just to go on the thought. I was asked yesterday a really important question. I was giving a talk to an organizational real world evidence summit, internal organization, and I was asked the question: give us an example where a study from real world evidence, observational study, changed your mind.
Right.
And it was very hard to answer that question, because what you just said was that, well, you need clinical subject matter expertise to inform a prior, and then you need a good study. Well, if it's going to be informed by your prior, does it mean that if it changes your mind, that study was wrong? Like, is it not possible then for an observational study to change your mind, or is it just to confirm your priors? Which is a really, really interesting question. And I think the answer is that I think they still can change your priors. Ethan Weiss on his podcast talked about this story, and it really resonates with me with this peanut allergy story. This peanut allergy issue, which is, my oldest daughter has a peanut allergy, and we withheld peanuts from her for her first 2 years of life, because that was the recommendation from the American College of Pediatrics, Academy of Pediatrics. And that was, of course, plausible. Don't give kids stuff and that they're allergic to, because it might hurt them. But this smart person made this observation, an observational study, that kids in Israel didn't have as much peanut allergy as Jewish kids in the UK, and did an observational study and really attributed it to this early consumption of peanuts in Israel. And then it was confirmed later on with a randomized clinical trial that had an enormous effect size published in the New England Journal. So that was a story where an observational study, I think plausibility could have gone either way there. Certainly an observational study led to the overturning of a practice, ultimately confirmed by a randomized trial. But it is true that it's rare that observational studies really turn a clinician's mind, change their mind.
Right. And it's part of the problem in general with doing research in some ways, right? Because you need equipoise to randomize, or you need equipoise to, I guess, even pseudo-randomize in some sense. And part of that equipoise automatically impregnates whatever difference you're doing, whatever different things that people are doing. It means that there probably isn't going to be a very large difference between two groups, right? Unless you can get in really quickly, where nobody knows anything about
Yeah.
whatever, about the therapy, and there's no information about it. So unless you have some vacuum of knowledge that you can have people in, and you can do a trial. So you always have this, the event rates, or the differential event rates, are necessarily seem to be very small. And you touched on the fact that, cardiovascular therapeutics, just simply because we're getting better, it seems like the absolute events rates are low as well. But isn't I mean isn't that isn't that a bigger rate limiting step to coming up with these strong answers? In that are we perhaps looking for tiny differences? So whether or not you're doing RCTs and spending a bajillion dollars, or whether or not we try to do something that spends less dollars, will we always be somewhat uncertain because all the questions we're asking aren't resulting in big time differential event rates? So one, it's hard to show, because it may be hard to show even whether you're randomizing or not, and two, you can never be super certain because that differential event rate is so small.
Yeah, I think, the problem of small effect sizes influences both observational studies and randomized trials. I think randomized trials are really poorly equipped to find very small effect sizes too. One should ask, what is the importance of finding very small effect sizes? I think that what we'd like to find are big effect sizes that we can make a difference. So that whole, the whole sort of, problem of small effect sizes has led to this exodus of investment in cardiovascular therapeutics, I think, and randomized trials in the space, particularly from pharma. There's huge pharma organizations that have historical significance in cardiovascular who have no foothold in cardiovascular anymore. So I think, you're raising like really important questions. Now, specifically, what is the roles of observational studies in finding treatment effects that are small? That's really hard to ask for. I mean, I think that it's not well equipped to do those things. You can get enormous sample sizes and get tremendous precision, but if you have bias, then you'll find small effect sizes that are biased, and they might not be meaningful. So what I think that observational studies are quite good for, and we're doing some of this work now in conjunction with both industry and the FDA, is they are good at ruling out potentially large harmful effect sizes. I think that's useful, and they're good at ruling out like sort of overstated benefits as well. So there are a couple examples I'm trying to think of. A couple examples I can give you. One is, so we're there is a safety concern about paclitaxel coated balloons used in peripheral vascular disease. So this is a drug-coated balloon. It's used to, open up an artery in the leg and prevent its recurrence from blocking up again. And there was this meta-analysis based on a moderate number of, I would say, rather poorly conducted randomized trials with much loss to follow. But this meta-analysis suggested that there was this harm signal for this device. The FDA wasn't sure what to do about this. But we can look in hundreds of thousands of patients. We have done this, and we're continuing to do this as part of analysis that we're calling the SAFE PAD study, which is co-led by my colleague, Eric Sosemsky. And in hundreds of thousands of Medicare patients, we can rule out, I think, we have looked at this and we're continuing to, but at least thus far, the preliminary analysis does not show any evidence of harm, mortality-related harm associated with the device. Fairly narrow confidence intervals. And here we're not looking for benefit. What we're really looking for is the upper limit of harm. So that is a case where a large sample size is useful. And in this particular case, there doesn't look like there's that much treatment selection bias. So what you were mentioning, it doesn't look like clinicians are really leaving just the sickest patients to receive the non-drug and just the healthiest patients to receive the drug-coated device. That there's some randomness with which these devices are used, and it's related to formularies and access to the new devices in different hospitals, et cetera. So that case, I think it's been useful for this observational research approach to take place. And we've also done, we've proposed a sensitivity analysis using an instrumental variable approach. There are falsification endpoints that I mentioned in this analysis, where we don't expect drug-coated balloons to affect endpoints like heart attacks or heart failure hospitalization. So those are endpoints that are built in that we hope will be negative in the trial, because that will show that we're not confounded. So there are ways that one can look at these types of questions and hopefully make observational data useful. The whole concept of equipoise that you bring up, so obviously it's an area I know that we've had conversations about this, and it's such a really, really, really important area. And I think what happens is, if you do there's usually no equipoise within one physician. So within your practice, there probably is not a good comparison to compare people that you give a drug to versus that you don't give a drug to, because you are very carefully deciding in the course of your day, and looking at this patient, why is it that I want to give this drug? Why is it that I want to use this device, treat with this procedure? And why is it that I don't think it's right for this other patient? But there still may be equipoise in the community, because I might treat a patient entirely different than you, and I might use the drug or device 80% of the time, and you might use it 20% of the time. So if you did analysis within my patients, I don't think you would get a confounded analysis. But between our patients, we might get actually an unconfounded analysis, if that makes sense. And that's one of the things that we've tried to explain in some of these studies, which is what we think is random preference. Let's face it, there are things that some docs like and other docs don't like, and it's not always based in evidence. It's how you were trained and what you have available to you, and those types of randomness can be exploited.
So, some of the studies that have kind of hit the that have made a little bit of a splash. For instance, a lot of studies by Dr. Jenna, right? Jenna.
Yeah, good friend of mine.
Yeah, exactly. So I'm not surprised. So, like the study on cardiologists that go to conference, and whether mortality changes or not. Now, so are you moved, or in their marathons, how mortality, there's some signal, correct, that exists? Is it ever, Bobby? So you come out with these, and it's somewhat binary. You come out with a conclusion that says, yes, mortality goes down, yes, mortality goes up, or do people ever look at data sets, think of answers that they could come up with, and conclude that this is too messy and we should not even try to do this?
Yeah, often.
Or is it, or why? I guess I'm also asking, why is it that Dr. Jenna has to put out this stuff, and then it's up to me to try to figure out why this doesn't fit my prior? Then I feel like, all right, well, what type of analysis did he do or whatnot? Why is the default, someone crunching some large data set and coming up with some answer, and then it being left to me to disprove?
Yeah, I mean, it shouldn't be the default. I don't know that it is the default, but it shouldn't be. I mean, I think what you're saying is, why is it that somebody who's outside of the field onlooking can do a study that influences your practice, say, and then left for you, who is actually doing the work of treating a patient and needs to make the actual decision being evaluated, it's left to you to sort of determine whether or not you should believe it or not believe it. Am I interpreting it right?
Right. Yeah. Right. Exactly. Yeah. But
this gets at the question. I think, I don't know if did you write something today, which is who would you want in charge of health policy?
Yes, exactly.
You wrote that today, right? Like, do you want a health policy wonk, an economist?
Yeah, who should decide physician value? Is a health policy wonk, a non-physician, a non-physician researcher, or an economist?
Yeah, and this is like, so who should do observational research? Is it someone who's seeing patients, or someone who doesn't see patients? And so, as I sort of alluded to earlier, and said out loud, is it has to come. The questions that influence medicine have to come from medicine. Right. And to me, sometimes you see papers that are clinical questions that have no clinicians on those papers, and that's a recipe for disaster. That's a recipe to get a totally implausible answer, or an answer that has no relevance to clinical care. So I think that it shouldn't be that it's just and I'll say, for Babu Jennas' case, Bob is a practicing clinician, so it may not be relevant to his science, but there is science that happens which is devoid of clinical input, even though it's meant to answer clinical questions. I think that's a problem.
Yeah.
So it should come more
Right.
from people who are actively making the decision. Now, there's this other thing, which is, I think, that you're also touching on, which is, what questions should we be asking? And so the field of economics is interesting because it's diverged from the field of biostatistics and epidemiology or clinical questions, because what the questions that you and I want to know are the ones, like, we have pressing questions that we're trying to answer because we have a patient that we're trying to make a decision for. But that is not always, that's not the best way to, those questions are not often best answered by traditional kind of, or these new kind of quasi-experimental methods, because natural randomization or quasi-experiments are hard to find. So there may not be a natural experiment for Impella versus no Impella when you're trying to compare that. There just happened to be like a nice random experiment for cardiology meetings or marathons, and those might not be issues that you or I are thinking about or caring about in the moment. They can be cleverly answered by an interesting method, but they're not the pressing questions that we are facing. We actually have a directed question, not like, so I think that there's this, the way that somebody, I think, had a really sort of well thought out thread about how the fields have come to be, and what he described, and I'm forgetting his name right now, is that economics has said we shouldn't even tackle questions that we can't answer well. What we should, but there are sets of questions that we can answer well, and then we should answer them. And epidemiology and bio and clinical questions have said, well, there are questions that we need to answer. So whether or not we can answer them well, I'm going to put that aside. I'm going to answer them to the best of my, and sometimes, but sometimes those questions shouldn't have been answered in the first place because they're too confounded. And it is rare, you're right. I've been a part of a couple papers where I've thought, I just don't think that we can answer this question, even though we're knee deep in it. And there's a trainee first author who really wants to publish it. There might be some other investigators who really want to publish it. And we have put the brakes on a number of publications because we said, and I'll say secondarily that I've taken my name off another handful of publications that ultimately were published because I didn't think that those comparisons were valid or should go forward. I don't know how often that happens, but I don't know that it happens a lot.
Right. Yeah, no, that's wonderfully, and certainly not surprising to hear from you. But there's no question. We'll wrap up here because I'm sure you're super busy. I think there's little question that clinicians doing this type of work is so incredibly important, and having folks that don't have some depth of knowledge of the field doing the research, I have a much tougher time, shifting prior based on an economist who starts talking about physician value, for instance, versus say your group, right? So if Rishi Wadera or you, put out a paper, saying something, certainly we're going to take a lot harder look at that. So it's almost like, shifting priors becomes about trust in the people doing the research, right? And so I think that's really incredibly important, and I think it's very important what you guys do, what you do. So please, keep it up. The other interesting thing about this work is that is that we've seen certainly in econometrics. When I was a much younger man, I gobbled up econometrics, Freakonomics, sorry, and Freakonomics is basically kind of a tome to econometrics, and you had these, and the kind of questions they were asking and answering were just like remarkable, right? They were talking about crime rates dropping nationally in the 1990s, and they were using these methods to say that it was because of Roe versus Wade passing in the 1970s, with all the implications of that. And I just gobbled them up at the time and just kind of swallowed it and kept moving on. And then later on, only when it kind of touches your fields do you start to really question some of this stuff. So I think it really points to having some trust and having in the folks that are doing the research. And then as as others have put it, Dr. Leamer talking about econometrics as well, puts it is, someone has to be able to give you some type of a compelling story, and you have to kind of, um, you know, you have to kind of be able to buy in, buy into it. So,
Yeah.
but
I always say this, which is like expertise is not only is expertise local, it's like hyper local. When I say that, I mean that, you can be just a hair's breadth away from a field and not have the least bit of insight, unfortunately, into the things that drive that field. And, you know, and so as like a non-invasive cardiologist, or me as an interventional cardiologist, actually, even just thinking about cardiac imaging in a study, I just don't think that I can bring the same
right,
expertise to evaluating that study. But that difference is sometimes what it takes to identify a problematic study versus not.
right,
Um, and the flip side of that, though, is that being too deeply embedded in a field is it's hard to remove yourself from your own biases. We can be led astray, and so it's that conflict, I think, that is really challenging to sort of navigate. It's like you have to be really deeply within a field, but sometimes you have to maintain in your own mind the skepticism or the ability to sort of have your priors overturned, um, but to provide the expertise to do the analysis correctly. um, that's like a perfect case
yeah,
scenario. Now we have people who are just way far outside
yeah.
outside of your field trying to do analyses, and it's going to be deeply problematic.
It requires a tremendous amount of humility, I think, and that's one of the places where Twitter and social media has been, I think, so helpful, right? Because you do get such a massive variety of opinions. So in order to get to the truth, I'm really convinced that you need this savannah of people with all these different priors, right? You have in cardiology, you have a non-invasive guy, you have an interventionalist, you have an EP, and everyone is kind of chiming in with opinions with their own little background, and then you know hopefully the mass of folks kind of coming up with
Yeah,
good stuff. And
the wisdom of the crowd, I
right,
guess.
there's some crowd, absolutely. So anyhow, thank you, thanks so much for coming on and spending an hour of your super precious time. It was
Thanks for
really
having
really
me, Nate.
great.
Good to chat with you again.